Humanity was hit by a shocking wave in December 2019, and this wave brought the world to a standstill in the early parts of 2020, and that is the COVID-19 pandemic. This show, the COVID-19 Diary, an initiative of the Association of African Universities, AAU, and the National Research Foundation South Africa, NRF, seeks to unpack all there is about the pandemic. And in this first episode, my panelists and I will be enlightening you, our cherished viewers, on the origin of the SARS-CoV-2. You can join the conversation on our social media handles on Facebook and YouTube at the Association of African Universities. I'm Joan Akua Iyoha, and this is the COVID-19 Diary. Welcome back, and this show is by the kind courtesy of the COVID-19 Africa Rapid Grant Fund, sponsored by the National Research Foundation South Africa. You can join the conversation on our social media handles on Facebook and YouTube at the Association of African Universities. In the studio with me today are my colleague science journalists, and I'll allow them make their own introduction. And I'll be starting with my gorgeous lady by my side.
Thank you very much. My name is Isabella Tetteh-Ahinakwa, and I'm a producer and host for Health Africa. Health Africa on AAU TV is a health show.
Welcome.
And my name is Agyemang Okyere Darko. I'm the host of AAU Talks on AATV.
Yeah, welcome to you too.
Thank you.
Today we are gathered here because of the pandemic, the COVID-19. And I wouldn't want us to just go straight into the conversation like that. So I want us to lay a very, very brief foundation on viruses and diseases, because growing up, I for one, I thought viruses and diseases were one and the same. You hear that you're sick with cold or flu or whatever, but later I got to know that one causes one or one is caused by one. So Isabella, I want you to give us a brief definition or just a brief description on what viruses or a virus is or are.
Thank you very much for the question.
Yeah.
Now, when we talk of a virus, I'll first explain how the name came about.
Yeah.
Now, the name was coined by a scientist called Pasteur, and this was from a Latin word which means poison. Exactly. Now, when we talk of a virus are composed of proteins, and these proteins actually helps the virus to bind to a host cell. And each virus has one genome. When we talk of genome are a set of genes and genomic materials found in cells. Okay. Now, each virus has one. It's either RNA or DNA. RNA is ribonucleic acid, and DNA is deoxyribonucleic acid. Now, what these two does is the DNA stores and transfers genetic information, and then the RNA actually, the RNA converts the genetic information stored in the DNA. Okay. So a virus has either one of them. It's either the RNA or the DNA.
So you can't have both?
No, it can't have both.
Okay.
It's only one.
Okay.
And also, most viruses, I told you, they are composed of proteins.
Yeah.
Now, they are surrounded. In the virus is the RNA or DNA, and they are surrounded with the protein. But most viruses has other envelope covered around it. Okay. This envelope are also, they can also be a lipid membrane. Lipid membrane is a fat.
Okay.
Yeah, that is why when the COVID-19 came, we were asked to wash our hands with soap, so that at least it can kill the virus before it gets in contact with your eye or your mouth. Exactly. And viruses are actually infectious disease, infectious agents, just like the bacteria, fungi, and then helminths.
Oh, okay. Okay. Agyemang, she highlighted one, bacteria. And I know bacteria and viruses, they are quite, we hear of them most at times. I want you to help with the distinguishing between a virus and a bacteria, just a quick one.
All right. So we're going to do this by placing them on some metrics, survival, reproduction, and also their sizes, basically. Let's start with the virus. A virus basically is a non-living organism compared to bacteria, which is free living cells, which have all components perhaps of a single cell. Now, what happens with a virus is that viruses are non-living. They can't live outside the body, but they can only live inside the body when they get attached to a healthy cell, basically.
The cell has to be healthy.
And even when they are in the body, if they're not in the cell, they are dormant. Until they get in touch with a healthy cell, they still are dormant. Unlike the bacteria, which is self-sufficient, can produce its own food, can produce its own energy, can reproduce and survive because of what happens we call the binary fission. This is what enables bacteria to be able to, on its own, inside the body or outside the body, produce their own food. In size-wise, viruses are submicroscopic. They are very little compared to bacteria. Bacteria are quite bigger than viruses. If you look at the average size of a virus, 0.02 to 0.25 microns. And this can only be spotted under a high-resolution electron microscopic tool, compared to a bacteria, which can be perhaps measured in between 0.4 micron, that is like 1/1,000,000th of a meter of a diameter in terms of size. So we know that bacteria is bigger than a virus. Look at the mode of infection. If you're infected, let's say a virus infects you, viruses are much more more of a sinister, imposter stuff. They are systemic. When they infect you, they grow in size and they encircle. So if, let's say, they get infected to a cell in your lung, or let's say in your nose, they now would spread all over the body. Unlike the bacteria, that is much more confined and localized. When they infect, let's say, your one part of the body, they stay there. They don't multiply to other parts of the body, basically. So I think if we weigh the two, these two are all disease-causing organisms, all right? But compared to the degree of effect, I wouldn't want to take one from the other. But they also have good size, though. For example, bacteria in the body can play a role in the digestion and also decomposition. If you leave your food in the refrigerator and then you come back and then it's getting spoiled, that reaction going on is a bacteria in action or toxins. Compared to a virus, have a virus in your body also killing undesirable bacteria in one way or the other. Even though they all call disease, all right, but this is just a thin line between these two, bacteria and the virus.
And I think my research, I also realized that it seems viruses can infect bacteria. Scientifically. Isabella, would you want to add?
I would like to add up to the differences, actually. And as we said, as my colleague said, the virus is a non-living cell as compared to the bacteria. The virus also do not grow. They cannot grow. They rather multiply when they attach to a host cell. So instead of they growing to become big, no, they rather multiply.
Drink smaller sub.
Exactly. And also because the bacteria is a cell, a living cell, it has ribosome in it. But the virus does not have a ribosome.
Still on you. We are moving ahead. I'm just trying to wonder, because we hear names like some names of viruses like the Marburg virus, the Ebola virus, the HIV, the influenza, and some of the names are quite, I won't say funny, but I just, I'm curious to know how these names came about, or how they do their namings.
Now, according to research that I made, previously the World Health Organization used to name a virus based on the location it was first discovered, and also the scientist who discovered the virus. Previously, that was how they used to name a virus, but then they decided to stop because to avoid discrimination. I will never be happy if Ghana is named, is tagged to a virus like Ghana virus.
First found
I wouldn't be happy. I'll be discriminated. So they actually stopped to avoid discrimination against people. And now they rather look at the features of the virus to name the virus. When I talk about features, I'm talking about the structure of the virus, the shape of the virus, the kind of genome it has. It's either RNA or DNA. You get me? And also the kind of protein surrounded, whether it has a lipid membrane or it does not have. It's a naked protein. So these are some of the features they look at to name the virus instead of tagging it to a location.
Or
they rather
a
look at the features
or something.
Exactly.
Oh, okay. Agyemang, would you help us with the WHO's contribution with regards to naming the process?
Sure. So I believe the WHO, the World Health Organization, made a deliberate effort to ensure that to prevent these wrongful, perhaps negative impactly naming of the viruses, let's create a committee. So we had scientists and virologists all over the world coming together under the umbrella of the WHO to ensure we create the International Committee on Taxonomy for Viruses of virologists, to ensure that they sit down and they understudy these viruses as they emanate. And then, as my colleague mentioned, studying the genomic features and structure, we come up with a name that matches it and not the locality or the people or the culture where it comes from. Sure.
Yeah, I think that was a very good move by the World Health Organization. So we've been able to lay a foundation on viruses, the naming and all. So we want to move into the reason for our discussion today, and that is the coronavirus.
Sure.
I'm still on you, Agyemang. I want you to help us to talk about the origin, the meaning of the coronavirus. So you'll start from the family of 7 down to the 4 and the 2 and all that. I want us to know more on this coronavirus.
Sure. It would interest you to know, and also our viewers watching us, that we have a lot, hundreds of coronaviruses all over around us. And these coronaviruses are spotted or perhaps have their reservoir in animals. So they are known to be zoonotic. Basically, these are class of animals. We have the camel, the pig, the cat, and the bat. These 4 perhaps are noted to have the viral reservoir in them for SARS or coronavirus. Now let's begin with the very first one, which I would say we have 7 main coronaviruses, out of which 3 are very deadly, and the others quite mild and then moderate. The mild ones, perhaps, which can bring you perhaps common cold, not really deadly as such. You have very funny names. I wouldn't hold you to it if you can't remember the names. The very first one, you have what we call the 229E, the OC43, the NL63, and also the HUK1. These 4 perhaps are some of the very lightly, mildly, moderate strains of this coronavirus. Then we come to the very, very deadly, perhaps with very mysterious infection symptoms, which are the SARS, the severe acute respiratory syndrome, and then the MERS, the Middle East respiratory syndrome, and the novel SARS-CoV-2, severe acute respiratory syndrome 2. Let's begin with the SARS. In between 2000 and 2003, there was a virus, a coronavirus that rose, that caused some common cold, influenza, some of the symptoms. And scientists did their best to understand the virus and see its origin, where it came from, which animal bore the reservoir. And they realized that it was from the bats. It caused some sort of distractions all around as usual. Then later on, 2012, September, another COVID-19 strain was also discovered, which was the Middle East respiratory syndrome coronavirus. And you might say, why was there a Middle East tag to it?
Exactly what
I wouldn't blame them. It's Middle East.
It's Middle East.
There's no country called Middle East.
It's true. They are not really specifying it to a country.
And all of these were quite epidemic. They were just in that particular region. They didn't pan forward to be a pandemic. So it tells you that the weight of its effect was lower.
It was light.
I wouldn't say, surprisingly, in 2019, in Wuhan province in China, there was discovered another strain of COVID-19 or coronavirus, which is the severe acute respiratory syndrome coronavirus 2.
So it's like a build-up of the SARS-CoV.
Exactly. You can't compare it because there's a difference That is a whole new character, a whole new behavior, a whole new mysterious lifestyle. And this virus actually caught scientists to be on their feet for a very long time, because understanding the SARS-CoV-2 took a lot of time. It evolved over a year.
Wow.
As stands, we're understanding its character, its transitions, and its source. But let me just take you back to the mess.
Yeah.
The mess, after studies, what was realized that it was also having its reservoir in the camel.
Wow.
A camel species called the day-drone camel. And this SARS-CoV-2 also perhaps is also having its source in an animal. And I'll wrap up by saying that we have several conspiracy theories saying that the virus is engineered, but scientifically it has been proven that naturally these viruses can be transferred from the animal, which holds the reservoir, to a human being through a process we call the spillover event. As we interact with these animals, it can be through eating them or through being with them. There can be a spillover. And that is where these viruses can be transmissible and transferred from animal to human beings. So I think that has been proven that it's natural. Yes.
Then
And SARS-CoV-2, there we are, different in attitude, different in symptoms, and also deadly compared to the other ones, but much more deadly.
Yes, and that's why it's called the novel.
Exactly.
Because it's new and deadly, and just the build up on the interaction between animals and humans. I would want to align with you, because I think recently there was this outbreak of the bird flu in Ghana, and they interviewed, the farmers, and they pointed out that, fine, it's in the bird. If there is no interaction, by the time they go try to get one to sell, the birds definitely would start having some discharges from their mouth, and it would get contacted by the farmers, and the farmers in turn would interact with their family members, and the family members in turn will interact with market women and all. And that's a really good way of it being transmitted. So Isabella, we are now streaming it down to the new one, the SARS-CoV-2. Just help us with the COVID-19. How did they come about the COVID-19?
Right, before I move to the SARS-CoV-2, I also like to touch upon whether it was man-made or lab-made. Now, scientists say that when they look at the backbone of the virus, it has natural features. On the spike protein, even the spike protein tells that it's natural. And then on the spike protein is a receptor called the receptor binding domain, that is a RBD, that is also natural. So these are some of the features that tells that the virus was not man-made. People were saying at the beginning of the pandemic, people were saying that, oh, it's a scientist who went into the lab and then developed the virus and then spread. No, but looking at the backbone actually tells it is natural. Now coming to SARS-CoV-2, as my colleague said, SARS-CoV-2 was termed novel because it's something new. It's now being discovered in human. As he said, the first two, the SARS-CoV and then the MERS-CoV, actually did not cause severe symptoms as compared to the
SARS-CoV-2.
And then the first case was discovered on December 2019. It was reported to the World Health Organization in December 2019. Now, and then the name COVID-19 is actually the disease caused by SARS-CoV-2. So the virus is the SARS-CoV-2, and then the disease is the COVID-19. I'd like to explain the name COVID-19. The COVID-19 is coronavirus disease 19. CO is the corona, and then the VI is the virus, and the D is the disease. Then the 19, and the 19 is the year it was discovered. That's the 2019. And then on 11th February 2019, that was when the World Health Organization declared the name of the disease, that is COVID-19. And then first it happened in a country, which was China. And then on 11th March 2020, it was discovered, or it was declared as a pandemic, which was worldwide. Yeah, that was 11th March 2020. That was when the COVID-19 was declared as a pandemic.
Still by the World Health Organization. We've been able to lay that foundation and speak on the COVID-19, but we'll go on a quick break, and when we return, we'll move to the transmission process and in more details. This is the COVID-19 diary, and we'll be right back. Welcome back, and this is still the COVID-19 diary, and this show is by the kind courtesy of the COVID-19 Africa Rapid Grant Fund, sponsored by the National Research Foundation South Africa. And I still have in the studio my panelists Isabella and Agyemang. Welcome back.
Thank you.
Yeah, we've seen a very vivid ideal description of the coronavirus from its origin and the naming, and you touched on the structure of the SARS-CoV-2, the SARS-CoV-2. So I want you to explain in detail the structure. I can see a diagram there, so I want you to help us with the diagram, using the diagram.
Yeah, so Isabella and I will be doing this together in terms of the structure. She'll be giving more of like a detailed background of where it came from with the name and all that. I'll just be doing the talks to buttress her point.
Yeah.
So we see something very weird looking over there. Quite weird.
Yeah.
You see it. It has some things around it. These are called the spikes around them, and it's using these spikes to gain access into the host cell. And then we have, you know, that's kind of like wormy looking, like a long worm, guinea worm in there. Yes. So that's actually the RNA viral genome, the real thing. The riboxynucleic acid viral genome. That was causes the virus itself.
I see.
And then it has been camouflaged. You see those dots around it.
Okay.
The black dots are what we call the nucleocapsid protein, which is the camouflage. It deceives the RNA in the host cell that it's protein. Because it's just around it. It's the camouflage. Without it, it will be spotted to be a virus. But with this, you wouldn't see the virus. So the ribosome will be just, I think she'll talk about that. How go there far. Then we also have what we call the membrane, which also helps in the replication of the virus quickly when it's spread out, when it's replicated or translated in the host cell.
Isabella really
it helps to form out quickly. Then the envelope protein also there as the cover. So there we have it in its structure. This is a genomic structure of the severe acute respiratory syndrome coronavirus 2.
So those are the spikes, and it's that's one worm-like thing that goes into the body
that
after
is the RNA viral genome, the messenger virus itself.
The messenger. So Isabella, we have it's in a human body now. The virus is in a human body. I want you to help us with the transmission process and the symptoms when it's contracted. What are some of the symptoms?
All right. The SARS-CoV-2 is transmitted in droplets through the air. So, for example, I'm infected and then I sneeze, cough, or even talking to you, and I'm not in a nose mask.
And I am not in a nose mask.
Exactly. In the air are droplets filled with the virus. Now, you are also not in your nose mask. You are breathing in oxygen. Actually, the virus is going to go straight into your mouth, your nose or your mouth or your eyes. Now, the virus looks for host cell. Now, this host cell, it actually infects host cells from your throat down to your lungs. Now, I like to go to the board and then explain into details. Now, this is the transmission process. This is the virus in your body. Let's say it's in your throat here, going down to your lungs, looking for a host cell. Now, this is the entrance of, I'll say this is the surface of your host cell. On the surface of the host cell is a protein or an enzyme called the angiotensin converting enzyme 2, ACE2. Now, this enzyme actually helps to bind with the virus coming into your body. So this spike actually connects to the ACE2 and then it's attached together. Now, around the ACE2 is an enzyme called protease, and what this protease does is it cuts the spike protein of the virus, allowing the RNA to pass through the spike, through the ACE, ACE2 into your cell.
Wow.
Now this is your cell. This is inside your cell. The RNA passes through the spike protein straight to the ACE2, then into your cell. Now, in your cell is a molecular machine called ribosome. And what this ribosome does is it binds with the RNA that is inside your cell. It binds with it, and then in a process called protein synthesis, it produces viral proteins and then the viral RNA. What I mean is, when it binds to it, it actually produces. I told you that the virus does not grow; it rather multiplies. So you see that the ribosome is actually producing more viral proteins and more viral RNA, and this comes together to form more virus in your cell.
More of this.
More of this. Just one entered.
Yeah, but
and then through the protein synthesis, it produces more virus. Now your whole cell is infected, destroyed. Now the virus, the multiple virus will come out from your whole cell to attack another cell. Exactly. So that is how the virus is transmitted. Now I'll talk about how when you are infected, the symptoms of the pneumonia, and I'll use that with the help of the respiratory system.
Okay.
Now, normally this is when you breathe in oxygen, air passes through your trachea freely down to two tiny tubes called the bronchi. Now down to tiny tubes called the bronchioles. Okay. Now at the tip of the bronchioles are tiny sacs called the alveoli. Forgive me with the name. Yeah. Now what this does is when you breathe in oxygen, the alveoli expands. Okay. Because there's air.
Yeah, so it's
and now when you breathe out carbon dioxide, it's kind of compresses. Yeah, and these are done with the help of blood vessels around the alveoli called capillaries. Okay. Now you are infected with the virus, and in you is your immune system or your immune cell trying to fight the virus. But then if you have existing disease like you are diabetic or hypertension conditioner, your immune system is kind of weak and is not going to fight the virus as it's supposed to do. Now when this happens, you see that the virus is in your alveoli and then your immune system is trying to fight it, but then it can't. So you see that dead cells will begin to fill the tiny sac, and then fluids, the mucus, will also be filled in your alveoli. Now, instead of air being in it, it's rather filled with exactly, and then the dead cells. Now, your sac is filled with those things, and you don't have enough air to breathe as enough air that your body needs to breathe is no more there. Now, you see that you'll be suffering from shortness of breath, pain in the chest, and then it develops to the pneumonia. And then if you don't take time, you actually die. So this is how you develop your symptoms, and this is how the virus actually transmitted from one person to another.
Wow, thank you for the diagram, and it's really well explained. And I can really understand it better now, especially when it's blocked, and that's when it's critical, and then they need a ventilator to aid the breathing. So Isabella made mention of the ACE2 in a description, the diagrammatic description. And with the way she said it, seems it plays a very, very important role in this whole process. So I wanted to talk on that in detail, the role of the ACE2 in this process.
Great. Yeah. Let me use the opportunity to just highlight the role of the ACE2 and why it plays a very critical role in ensuring that the virus enters or perhaps infects the whole cell. Now, ACE2, which is known as the angiotensin converting enzyme 2, is like a protein on the surface of many cell types, like the liver, the lungs, the bloodstream, the blood vessels. Even in our respiratory lineup, there is an aspect of ACE2. What ACE2 does basically is to ensure that it regulates blood pressure, healing of wounds, inflammation.
Very vital.
Very vital. And this same ACE2 also, as an enzyme, break or cut down large sizes of protein into angiotensinogen, which is smaller sizes of protein, so they can also take part in the functional role in the body. Moreover, this same ACE2 is responsible to modulate the activity of a particular protein called angiotensin 2. Now, this protein called ANG2, which is the angiotensin 2, has a quite defective role or function.
Okay.
This protein is responsible for increase in blood pressure, inflammation in the body.
Okay.
In the cell, basically.
That's the angio-2.
ANG2.
ANG2.
And ACE2 is responsible to ensure that as it does its job, it puts ANG2 in check.
Okay.
So if ACE2 is dysfunctional, ANG will go haywire.
Yeah.
So ACE2 needs to be focused and more comported to ensure that it places ANG2 in check. So why then would there be, as my colleague mentioned, that it gets to a point whereby your lungs begins to flame up, filled with mucus. And when air inflates these lungs, they can't deflate because there has been an overtake of mucus. Why? One of the symptoms of coronavirus and its effect to the lungs is that it inflates the lungs, bursts open the capillaries, affects the alveoli, and turns the whole place on fire, burning, inflaming your lungs and making it hard for it to take part in the air exchange of carbon dioxide as you take in oxygen. What happens then? Now, when the coronavirus, because it has a spike protein, which deceives the ACE2, that there's a protein approaching. So allow it entry. It allows entry. The protein does its job because it believes it's protein. It's a camouflage in there. RNA viral genome, which is also camouflaged with nucleocapsid protein, will now make entry into the host cell. Now, this coronavirus, as very mysterious and very important as it is, does not even need to get to the nucleus of that host cell. It just will hang around the outer layer, which is the ribosome, and that's where it gains a chance to gain translation. With all this effect, the SARS-CoV-2 virus will now turn the ACE2 away. So now you compromise ACE2 from doing its regular functions. That means that ANG2 will now be left alone. If you turn ACE2 away from its functions, because you've made entry into it, and then you compromise it, then ANG2 won't have any enzyme to put it in check. So now it will do exactly what it does always, without modulation, cause inflammation and blood pressure. And that is how come the body, after some time, will begin to experience such very hideous symptoms of inflammation of the lung and other symptoms, because the ANG2 has been left alone to cause damage. That is the trajectory of the damage coronavirus can cause, the SARS-CoV-2 can cause to your system if it happens.
It is quite surprising and an unfortunate mystery how something that is supposed to fight for us and produce something good now kind of turns against us and more like fights us. Yes, it's quite a mystery. Still speaking on the COVID, so over time we've seen the, in quotes, evolution of the lifespan of the virus on surfaces and all. I remember initially they said, and the surfaces vary. On a wooden surface, the lifespan is quite different from a metal surface on dresses and all, and if you are exposed to the sunlight. Over time, it started going to like 36 hours, 72 hours. I would like you to explain and it got to a time we also heard that it's airborne. Yeah, so speak on that briefly before we wrap up.
Well, scientifically, scientists have done their best to understand the virus, the SARS-CoV-2 virus, to an extent where scientists have actually brought out information that this virus can actually stay on some surfaces for some time. And these surfaces vary depending on the organic structure of these surfaces, perhaps the organic matter of wood, of metal, of, let's say, any other substance, and how it's made up and how it can accommodate the virus. You know, viruses, as we said in the beginning, are non-living and will be living if they get in touch with the host cell. And if that particular thing is not living, if a virus is not on a living material or a living matter, how can that virus survive? And for how long can it survive? They can't stay for long because it doesn't have anything that will help them to replicate or perhaps live. So let's, on the surface of a wood, you have science between, let's say, 26 hours to 72 hours. On the face of a metal or copper, you can stay like 24 hours. Variously, 24 hours is the least. A day is the least duration for a virus on the surface of the material. That tells you that if the least is 24 hours, then what will it be for the rest? Then perhaps you need to put yourself in check, because the least it can stay even on the basic surface is 24 hours. And the highest perhaps 72 hours. That is like 3 days.
The highest, 3 good days.
Sure.
In rounding up, I really appreciate your coming. In rounding up, I wanted to round up with preventive tips. Just share some preventive tips with our audience.
Now we know that looking at the structure of the virus, we know it's covered with a lipid membrane, which is fat. So it's best we keep on washing our hands with soap and then under running water. At least it's going to kill the virus. And if you're in a vehicle, there's no water and soap available, continue using your hand sanitizers. The 99% alcohol.
And there are portable sizes now that
it can be in your bag.
Exactly.
You just use it frequently to kill the virus before it gets in touch with your mouth and your nose for the virus to enter into your body.
Agyemang?
I think that now people or viewers now understand why 2 years ago there was an advice that do want to eat more organic foods and also do more exercises, drink more water, because, or let's stay home or wear your nose mask, because the genomic structure of the virus is such that it is much more it is immune to some food substances, and also perhaps not so comfortable with some food substances. So you need to understand the differences here, so you can align yourself well to the right place. So with your diet, I mean, if you know that now you know the virus is made up of proteins and fats. Therefore, perhaps you wouldn't be consuming more fats. You understand why you shouldn't consume more fats or more proteins, because the more you do that, perhaps you create that safe haven
for them
for the
to
virus
thrive.
to translate more. But when you go to more of a, taking, let's say, portfolio diet, which is much more vegetative, and also food fiber, plants, vegetables, fruits, then perhaps you don't give this virus the room to replicate or perhaps to overcome your immune systems. And also the exercise aspect. When you exercise, you burn you actually burn some cholesterol, which is also good for your body, because when you do that, you reduce the fat content in your body, also reducing the strength of the virus and giving more strength to your immune systems. This is the reason why every reason to a fact. Now the facts are now giving reasons to people to understand why it's done. And finally, stay home or wear your nose mask, because you have no idea at what point in time
it's waiting,
or what environment or what encounter would be coronavirus infectious. So when you touch the surface, you want to wash your hands, or perhaps sanitize, wear your nose mask, and then you are safe.
And in addition, try as much as possible to maintain a good social distance, and there are innovative ways of exchanging pleasantries now, like the elbow. You greet with your elbow, your feet, and all. It has been a fruitful session with my panelists, Isabella and Agyemang. And in our next episode, we will be touching on a very important aspect of the SARS-CoV-2. And this show has been the COVID-19 Diary, brought to you by kind courtesy of the COVID-19 Africa Rapid Grant Fund, sponsored by the National Research Foundation, NRF. I've been your host, Joan Akua Iyoha.